Hey guys, welcome to topic 2 in chapter 14. In this topic we're going to talk about energy generation in chloroplasts. This is partially photosynthesis a little bit, but we're going to really talk about how oxidative phosphorylation is used to generate sugar molecules and other molecules that can be used throughout the cell in the plant. So this topic outline focuses on photosynthesis and chloroplasts. Here are your topic objectives. As always, these are what I expect you to have mastered before our next exam. So if you have any questions, please let me know. Alright, so now let's talk about the chloroplasts. Chloroplasts and mitochondria are really similar. They have DNA, RNA, ribosomes, and they also have a double membrane, but their double membrane is really limited to the perimeter because what's on the inside is very different. What they have is this jelly-like substance called the stroma, and then they have these pancakes of little individual compartments called thylakoids. And these thylakoids are where photosynthesis will occur. So it's important that you understand how these structures are different. Whereas the mitochondria has all these invaginations forming cristae, the chloroplasts have thylakoids and they're in a different format. So here is an overview of photosynthesis. Now this topic is going to be really short and sweet over this, mostly because I expect you to have a lot of this already done. We're just going to talk about it in a few different details that we're adding to it. Based on our previous conversation we had about the electron transport chain. So what we have here for photosynthesis in the most simplest of terms is we take light, we harness light to break water, which is going to then create ATP and NADPH, which is going to go to the Calvin cycle for carbon fixation, where sugars, amino acids, and fatty acids are going to be made for the cell. And for the plant. And this makes sense because plants can't move, they can't obtain nutrients from anywhere else, so they have to be able to create everything they need. Now what I want you to do is I want you to pause this recording and I want you to write down the products and reactants of both photosynthesis and the and chemiosmosis, cellular respiration, whatever you want to call it, the processes we've just been talking about. Basically, I'm asking you to write out everything that's going on in the carbon cycle. All right, so you're back. Hopefully, your carbon cycle looks something like this, where we have the aerobic respiration is going to have the 3 stages of glycolysis, the citric acid cycle electron transport chain. They're going to produce high energy electron carriers, ATP, water, and in this case we have 26 to 28 ATP, but as we saw in the last topic, our book estimates it more at 30, and carbon dioxide. And these are going to go to photosynthesis. Well, some of it's going to go to photosynthesis. And this is where it's going to create this big circle, and these things just keep working, whereas photosynthesis creates oxygen and sugar, which are then going to be used in the aerobic respiration process. So it's really important to understand how the carbon cycle works, because this is what generates life on this planet. And really we talk about this a lot. We talk about all these processes and all these molecules and where things are going, what proteins are doing what. But if you don't understand this, we really need to talk, because the carbon cycle is what drives life on this planet. And so I really want you to understand how photosynthesis and aerobic respiration work together to create life, or create the means for life. And in topic 3 we're going to talk a little bit about how the chloroplasts and mitochondria have helped life develop on this planet. But really spend some time with this. Alright, so now let's talk a little bit more in detail about photosynthesis and how this works. And we're going to start with the light reaction. So photosynthesis has 2 phases, as we talked about. They have the light reaction and the Calvin cycle. The point of the light reaction is to generate NADPH and ATP for the Calvin cycle, because the Calvin cycle needs energy in order to build those macromolecules. And they also need NADPH to do that as well. So what's going to happen here is instead of using high energy electron carriers to drive the electron transport chain, we're going to use light to stimulate the electrons that have been donated. Now in this case the light is going to be in these tiny little packages called photons. And in green plants these photons are specifically, they're red and blue photons, and they're going to hit these molecules, these chlorophyll molecules that you can see within the thylakoid. And what it's going to do is it's going to bounce around. All this energy is going to bounce around until it reaches the reaction center. at which point in the reaction center the donated electron is going to become charged, and then it's going to be excited and transferred then to the next molecule that carries the high energy electron forward in this process. Now in this case, if it's photosystem 2, this electron comes from water splitting. This is where water comes into play, and this is where molecular oxygen is released. This is where the oxygen that you and I breathe comes from, is from photons hitting these sites, allowing the water to be broken, releasing the electrons, releasing oxygen out into the environment. And so you can see a little bit about how this harvest of light energy occurs. So now let's look a little bit more at the electron transport chain in plants. And it's a little bit different, whereas in the mitochondria, the electron transport chain's main job was to pump protons across the membrane. And in this case, we do pump some protons across the membrane because we need some to drive the ATP synthase. But what else we need to do is we also need to generate NADPH. And this NADPH is generated from photosystem 1. Now it's important you get your nomenclature right here because we start with photosystem 2, transfer the electron through the cytochrome B6F complex to photosystem 1. So we start with photosystem 2 and we go to photosystem 1, where the light will re-energize those electrons and allow for the generation of NADPH. Now because of the proton pumping that we've done, we then have ATP synthase that will generate that ATP. So make sure you understand how this works and who's pumping what, because it's different. It's not everybody's trying to pump protons across. We have one set that works on generating energy, one set that works on creating NADPH. Both of these products have to be generated in order for the Calvin cycle to occur. So now let's talk about the Calvin cycle. The Calvin cycle harnesses or fixes CO2 from the air and generates molecules for use in the cell. These can be sugars, they can be fatty acids, they can be amino acids. It's whatever the plant needs. And so what it does is it goes through a similar process like we talked about in the Krebs cycle, except for kind of in reverse. I'm sorry, in the citric acid cycle, but kind of in reverse, where instead of getting ATP and getting electron carriers out, we're putting energy in. Lots and lots of energy have to go into building these molecules because, as we talked about in the laws of thermodynamics, this is not a favorable reaction to generate these molecules. So all this has to go in order to continue to create these for the plant. The Calvin cycle can occur without light, sometimes called light independent reaction. I'm not too worried about that too much, but I just want you to understand what the purpose of the Calvin cycle is, what goes in and what comes out. That's really the main goals here. All right, so here's the products, and then once again a summary of what we have going on. Once these sugars are created, they have a couple different options. They can go and be stored as starch. We talked about the potatoes storing their energy stores as starch and potatoes, but they can also go and be used in the mitochondria for energy creation, because the ATP that's created in the chloroplast is created almost exclusively for the Calvin cycle. If we want to generate a lot of ATP for the production of energy for the plant, we need to use the mitochondria. Remember, plants have mitochondria as well, and that's because mitochondria is the powerhouse of the cell, while the chloroplast is essentially the exact opposite. Its job is to produce sugars, fats, and amino acids. It creates the biomolecules for the plant. So this is a quick summary and a quick discussion of how we use membranes to create molecules within plants, and how these cells work to essentially create or to substitute for what we do when we ingest food. So make sure you review your objectives and let me know if you have any questions about any of the items we talked about in this topic. And when you're ready, go on to topic number 3.